Good afternoon, everyone. This is Dr. Jeff Gelblum, your friendly neurologist coming to you live from First Choice Neurology, based down here in sunny South Florida. And thankfully, today is a sunny day. We've had some bad weather over the past few days, but today the sun is shining. So that always bodes well for an educational discussion like what we're about to have. For any of you who have seen the invitation for today's program, you know I always recommend two things, a pen and the back of an envelope, because with a pen you're going to write the important notes that we're about to share regarding today's topic. And as an old professor of mine once said, if you can't put all your thoughts down relevant to a certain topic on the back of an envelope, then you don't really understand the topic at hand. And of course, there's always the old expression, if I had more time, I would have written a lot less. So today we don't have a lot of time, but what I'm going to do is take you through the ins and outs of what you need to know about this hot topic called coronavirus vaccine development. So what do we mean by a vaccine? What does the word vaccine come from? Well, we have to go back to our old friend, Dr. Edward Jenner, who was a British physician back in the 1700s. And he understood the idea that milkmaids, meaning people, ladies who dealt with cows, did not get smallpox. And as you may know from history, smallpox was always a terrible affliction affecting mankind for thousands of years. And it was his observation that milkmaids didn't get smallpox. So what he would do is he would take a little bit of the liquid from the pustule of the cowpox milkmaiden, and he would take that liquid and he would inoculate that into someone who had never seen smallpox and never seen cowpox. And with that, he learned that people who had received a smallpox inoculation, people who had received a cowpox inoculation were immune to smallpox. And with that, we have the idea of what's called vaccination. Vaccination comes from the word cow, vaca. So the term vaccination goes back to the time of Edward Jenner, 250 years ago, where it was his observation that milkmaids who had contracted cowpox did not get smallpox. And he used that observation to then start to inoculate new individuals with the cowpox, and that created immunization against smallpox. So that's the history of vaccination. So basically what he did is he used one pathogen, the variola pathogen of cowpox, to create immunization against smallpox. So this whole idea of vaccination goes back 250 years, and here it is once again front and center for our confronting the coronavirus pandemic. So let's talk about what is required to develop or get a vaccine to market. And as with any other therapeutic intervention, be it vaccine, drug, medical device, it has to go through, at least in our country, the FDA, the Food and Drug Administration. In Europe, it would have to go through a European agency, and in Asia, there are other agencies, for example, China, Japan, etc. But for purposes of an American discussion, we have to talk about FDA approval, because the FDA would have to approve any vaccine before it could become available to us all. So what are the different phases of FDA approval? You're hearing a lot about phase 1, phase 2, phase 3, phase 4, something just passed phase 1 and 2 and is now in phase 3 clinical trials. So what do these different phases mean? Well, for purposes of the FDA approval process, there are basically 3 phases that you have to know about. So here's your first point that you have to jot down with your pen on the back of your envelope. The first phase is called phase 1. And what that looks at is you take your substance and you give it to a small group of people. And A, you see that the people don't have serious side effects, and B, you're able to figure out somewhat of the dose. So phase 1, small group, primarily looking at safety. Phase 2 is a larger group, meaning once that product has overcome the phase 1 evaluation, then you go into phase 2, which is a larger group of individuals that is a broader overview of safety, and you start to look at effects, meaning what are the effects of the therapeutic product that you're trying to achieve. So when it comes now to some of these coronavirus news flashes that you're seeing on the front page of all of our journals nowadays. Things are coming from phase 1, meaning small group has been shown the product and has survived and hasn't suffered any significant safety outcome as a consequence of receiving the treatment. And now they're going into phase 2. And phase 2 means larger group, and now you're starting to look for effects. So what would be the effects? Well, in an antibiotic clinical trial, you'd be looking for the ability of the drug to stop an infection like pneumonia. The recent studies that have looked at hydroxychloroquine were looking at effects of stopping coronavirus. Of course, all that data has been put to bed because basically nobody's looking at hydroxychloroquine anymore. But purposes of phase 2 is to look at larger group, safety encounter, as well as to look at the effects of the product. When it comes to a coronavirus vaccine, a COVID vaccine, what you're looking for is the development of antibodies in that human that has tried the experimental product. And with that regard, you're able to assess the effects of that product on the body, as well as its safety in a larger group. So if the product passes that hurdle, then we go to what's called a phase 3 FDA level trial, which means that now you're looking at a large group. You're comparing the drug to a placebo, to a sugar infusion or injection or sugar cube. And you're not only looking for safety in that large group, but you're also assessing response, meaning what are the overall response in terms of effectiveness, in terms of overall tolerability. So that's what a phase 3 trial is all about. And once something passes FDA level phase 3, at that point, you can say that the drug is going to be approved. So this whole process takes at least a year. So now that we're looking at the coronavirus phase 1, 2, and 3 protocols that are underway, how long will this whole process take? Well, here we are in May of 2020, soon to be June, and we can clearly say that we're not probably going to see anything in terms of a phase 3 FDA approval, at least until a year from now. So we're looking at least a year of phase 1, 2, and 3 clinical data that has to be looked at by the FDA, tested on a large scale to make sure, A, that the product is safe, and B, that it gives us the desired outcome. And when it comes to coronavirus vaccine, the desired outcome will be creation in the human body of antibodies that are neutralizing. So write that down, neutralizing antibodies. So it's all about the ability of this experimental product, this coronavirus vaccine, to be injected and to create the requisite, the necessary coronavirus, COVID-19 neutralizing antibody. Now, the studies that go into this are typically done with monkeys, guinea pigs, and mice, so that when the drug is developed, they inject these things to make sure that these neutralizing antibodies are created in the experimental model, which is usually a mouse, a monkey, or a guinea pig, because their immune systems are rather similar to the human immune system. So what that says is before something can even get to phase 1, the proof of concept is already there in the laboratory to see if this product has potential in the human to create that neutralizing antibody that will be required for coronavirus protection. So that's the history of vaccines. That's what we mean by phase 1, 2, and 3 FDA trials. Phase 4, which you might hear about down the road, is post-marketing surveillance, where the FDA goes in and sees and says, all right, we've seen 100,000 people take this product. Let's make sure that it still remains to be safe and effective. So that's what we mean by the term vaccine. That's also what we mean by the term phase 1, 2, and 3 FDA approved trials. So for purposes of where we are right now in the coronavirus vaccine space, the overall picture, we're looking at products that are emerging from phase 1, meaning been tested on small groups, and are now going into phase 2, which is looking at larger groups and making sure that there is the production of a neutralizing antibody in the human. So that's where we are right now across the entire spectrum of vaccine development. So now let's talk about what are the different strategies that are used for vaccine development. Well, you, me, most of us, we're not going to pay much attention to the anti-vaxxers. Hopefully, they're not logging on to today's conference because they'll always find fault with any vaccination approach. But for all of us who are on today's Facebook Live, we've all seen vaccines. And there's basically five strategies that have been around since the time of Edward Jenner for the development of a vaccine. So let's go through those five strategies, because as you read about these news headlines every day, then you'll hear reference to one of these five strategies. So what are the five strategies, and where have we been with these five strategies historically? Because from my perspective, history is often your best teacher in terms of what's going to happen and be feasible in the future. All right, so the first strategy that I want you to write down, again, you need your pen, you need your envelope. The first strategy that I want you to write down is what's called viral vector. Viral and vector, V-E-C-T-O-R. So what is a viral vector? A viral vector is a vaccine that is developed using another virus, an unrelated virus, and then the interior, the DNA, the genetics of that virus are altered to now create an immune response to what you're looking in the new disease state. So for example, the typical viral vector that is under investigation right now for protection of coronavirus, for creation of this viral vector vaccine set, would be adenovirus. An adenovirus we all get. That's a cold virus. So when you have a head cold, when you have a scratchy throat in the wintertime, that's an adenovirus. If your kid comes home from school, sneezes in your face, and you're sick 2 days later with a head cold, that's called an adenovirus, A-D-E-N-O virus, adenovirus. And we've used adenoviruses. We have manipulated adenoviruses in the past to create vaccinations. In fact, Merck did a good job of manipulating an adenovirus using that viral vector in its creation of an Ebola virus vaccine product. We use the adenovirus to create attenuated responses to other disease states, meaning we dampen down that response. So where are we right now? Who's got the hot topic right now? Who's got the frontline center product when it comes to manipulating a virus, an adenovirus, for the creation of a coronavirus vaccine, meaning they would take that altered adenovirus, inject it into you. Your body would view it as something very similar to coronavirus because the adenovirus has been manipulated in such a way that it looks like a coronavirus. So you inject that into your body. Your body says, uh-oh, this is something foreign, something strange. Your body creates an antibody response to it. And that is going to give you protection against coronavirus because the adenovirus has been manipulated in such a way that it looks like the coronavirus. So that's called a viral vector. Right now, the Chinese are the leaders in the creation of a viral vector vaccine, VVV. And basically the division within China that is creating that is actually an arm of the military. The People's Liberation Army owns this company called CanSino, and I want to tell you, for purposes of today's discussion, do not invest in any of the companies that I'm talking about, because I'm not an investment counselor, and as I always say, if I say to buy, it means you should immediately sell. So the top contender right now for the viral vector adenovirus transformed product that could become a coronavirus vaccination is the Chinese, and they've got this company called CanSino, or CanSino, I guess, for China. And what they've done is they've manipulated the adenovirus. They've used research coming out of Oxford University from a chimpanzee strain of adenovirus, and from that, they have created a viral vector that seems promising for the protection of the coronavirus infection. Johnson & Johnson, under its Janssen division, also has an adenovirus product transformed. Looks a lot like a coronavirus, but of course not as virulent, not as toxic. And they seem to be also favorable. There's another virus aside from the adenovirus, and I don't want to get too microbial or technical with this today because it is lunch hour and I want you to keep your appetite. Merck has a company, or I should say Merck is a company, and they've used a virus called the vesicular stomatis virus. And that's what they used in the Ebola disease state. So Merck has a good experience there. Merck also has experience with attenuated, with weakened measles virus, rubeola, and with that they're creating their own viral product that looks a lot, has a lot of the properties of the coronavirus and may have potential, but it's a capital May, M-A-Y in caps and underlines and lots of quotes. So none of these products has yet shown to be able to have broad spectrum immune neutralizing capability in the human population. And I want to remind you, here's the problem with attenuated adenovirus. Here's the problem with attenuated measles virus. We've all seen that. Meaning, I've seen it. I've had a cold before. I was vaccinated against measles when I was a little boy. So, therefore, the viral attenuation, the viral vector strategy may have historical basis, but the problem is because we've already seen this type of product before in our bodies, yes, we'll mount an immune response, but it may be an imperfect immune response. So, we won't necessarily get sick, but the virus will still be able to replicate in our bodies, meaning when we get hit with the coronavirus, we've already seen the attenuated adenoviral vector. We've already seen that in our bodies before. So if I get hit with a new coronavirus, I might not get sick, but my immune response may not be sufficient to stop the virus, the coronavirus, from reproducing in my body, and therefore I may be able to continue to shed fresh virus even though I'm not sick. So I may actually still be infectious even though I've had the vaccine, even though I've got antibodies, but because it's an imperfect immune response, because I've seen this type of viral vector before, because I'm having an imperfect immune response to it by virtue of a prior infection, because my response is not 100% controlled, at that point, I may still be in a position to create other viruses, other coronaviruses, and still remain an infectious risk. So that's viral vectors. We're done with that topic. There's a lot of research. The Chinese are front and center in that game. And I can tell you that there is historical predicate for utilizing viral vectors in the vaccine development arena. So we've got history there. And maybe with that history, we will have a coronavirus product. But for the time being, these products, these viral vector products, are still in phase 1 and phase 2 clinical trials. So we've just spent time talking about viral vectors, utilizing things like adenovirus, the head cold virus, utilizing things like attenuated measles virus. So that's where that research is going with viral vectors. So enough of the viral vector discussion. And I told you we were going to talk about 5 strategies, so we just did number 1, viral vector. What is number 2? Number 2 is creating, or I should say taking, parts of the coronavirus structure, finding the messenger RNA that makes that structure, and then injecting that messenger RNA into my body. And when my body now receives the messenger RNA, it's going to make the viral protein that RNA codes for, but it's not going to make the virus, just a component of the virus, and then whoosh, my antibodies come in and hit that viral component. So where are we with that? All right, well, we know very well the genetics of the coronavirus. We've had that worked out since December. So what the messenger RNA, the RNA Vaccination Strategy is looking to do is take the RNA that creates the spike protein, because you know the corona is a sphere with little spikes. That's how it gets its name, crown corona, meaning spikes coming out of it. The RNA vaccine strategy takes the RNA that is coding, that is making those spikes. We take that RNA, we inject it into the human, and with that, the human now makes those spike proteins, and we now then see this as a foreign entity, and we create lots of antibodies against the spike proteins. Has this ever been created before in a workable vaccine? No. However, there are companies out there now working on mRNA, messenger RNA vaccines that have the capacity to generate neutralizing antibodies in the human. And one of these companies that you may have heard about is called Moderna. It's a rather new company. It's never brought a vaccine to market. However, their technology seems to be front and center in terms of this RNA approach to taking the RNA that makes the spikes, injecting that into some humans, watching the humans develop the antibody to those spike proteins, and then watching the humans now be able to have those antibodies neutralize coronavirus when it's infected. In fact, last week, what caused the stock market to rise up hundreds of points was the fact that Moderna, this RNA vaccine company, said, hey, in the few people that we've injected with this messenger RNA, they made antibodies to coronavirus. So, wahoo, we've got the holy grail here. We've got the product that works. But I can tell you that has not been done on a broad spectrum basis. So that product, their version of the messenger RNA vaccine, is now going into phase 3 clinical trials, or I should say going from into full phase 2, larger group, and then we'll go into phase 3. So we've covered viral vector, we've covered messenger RNA vaccine. The front leader in the viral vectors is the Chinese, as well as J&J, Janssen, and Merck. The front leader in the messenger RNA strategy number 2 for the vaccine, is Moderna. And again, I'm not recommending that anybody buy stock in these companies, because it remains to be seen whether this stuff really works on a broad spectrum basis. So that's RNA. Now let's talk about DNA. So we can take RNA and inject it into the human body, the RNA from a fragment of the coronavirus, and see how that creates an immune response. Or we can take the DNA from a viral product. And the DNA from the coronavirus has been worked out exquisitely. There's a company out there called Inovio, I-N-N-O-V-I-O, and they've got their product, Inovio 4800, which is a product that takes the circular DNA from the interior of the coronavirus. That gets injected, and from that, the human body will create neutralizing antibodies against coronavirus. And you know what? In a small cohort of individuals, as of last week, that seems to be the case. And in fact, this strategy seems to work in guinea pigs, and I'll tell you, all the strategies seem to work in guinea pigs, but whether it actually works on a broad spectrum in the human population, still the jury is out. We can't say that this is a strategy that is actually proven historically, because it's never been done before, just like the RNA has never been done before. Attenuated, viral vectors have been done before in a viable product, but not RNA vaccines and not DNA vaccines. So the RNA story, the DNA story sounds extremely compelling, and the advantage to the RNA and DNA strategy, you might be saying, well, why are they going with all of these new strategies? Why not just go through the old tried and true method of either using a viral vector like we use oftentimes with the adenovirus? Why are they doing that? The reason why they're doing that is because to create a vaccine from RNA or DNA is rather quick and can be mass produced. So when you're talking about other vaccine strategies, time is always a limitation here. So therefore, everybody's trying to come up with the quickest strategy to create a broad volume of vaccine doses, because basically you're probably looking at the need for 6 billion doses, meaning we have to cover humanity worldwide. So that's why the RNA and the DNA strategies seem to be so attractive right now, because they can be quick strategies. Meaning once you have phase 3 trials that look good, at that point you can manufacture that stuff very quickly. So that's strategy number 3, DNA vaccine. We've talked about strategy number 2, RNA vaccine. And we began with viral vector strategy, adenoviruses, attenuated measles viruses. That's strategy number 1. So what would be strategy number 4? Strategy number 4 is where you actually take components of the virus, you manufacture parts of the shell, parts of the crown, parts of the interior. You manufacture components, proteins from the virus. You inject and you watch the immune response hit. Has that ever been done before? Absolutely. That's our annual flu shot. That's done with young women to prevent against HPV virus. So that is a tried and true strategy. Viral proteins that are stripped off of the coronavirus, you inject it, and with that, hopefully, you're going to have an immune response in the human. Now, somebody today texted a message. What about this company, Novavax, based out of my home state of Maryland? Well, what Novavax is doing is taking the spike protein parts of the coronavirus, and they're putting it on this super duper nano matrix technology. They're injecting it. And with that, hopefully, when your body sees those little particles floating around, it's going to create antibodies that should then be enabled to stop coronavirus when it hits your body. So that's the Novavax strategy. That's the viral protein strategy. That's something that's already in place when we talk about human papillomavirus, HPV, that then goes on to cause cervical cancer in women. And that's also a strategy that is used each and every year when you go and get your flu shot. In fact, when you get your flu shot, there's 2 ways to get a flu shot. There's the Sanofi product. Sanofi is a French drug company. There's the Sanofi product, which uses viral proteins from the H1N1, the influenza bug. They extract that. And with that, that gets injected, and boom, you get an immune response, hopefully against this year's flu. There's another strategy that is used for flu, and that's where they basically come up with an attenuated virus, an inactivated virus. And we're going to talk about that right now, because that's strategy 5. So let's recap. Strategy 1, viral vector, modulating, changing the adenovirus, a stomatitis virus, a measles virus to make it have components of coronavirus, inject it, and hope that you get your immune response. Strategy number 2, taking the messenger RNA that we know to exist in the coronavirus, synthesizing that, typically of the spike protein, injecting that, and then watching the body make lots of spike proteins. And once you make these spike proteins, your antibodies are going to say, whoa, this doesn't belong here. Create lots of antibodies against spike proteins so that when you inhale the coronavirus from the person who's not social distancing over there, hopefully you've got enough antibodies to protect you against that infection. The third strategy is, of course, the DNA strategy, where they inject the DNA from the coronavirus, and then your body makes variations of that, and then antibodies are produced in your body, and that's how you stop this guy who just sneezed on you from giving you coronavirus. So those are the 4 strategies, and then the last strategy that I always want to remind you that's a tried and true strategy is what we call the inactivated pathogen strategy. That's how we all got our polio vaccines years ago. They would take polio, the live polio virus, they would boil it down or denature it, and then they'd inject that into you. And your body would say, uh-oh, polio, even though it's a very weak form of polio, and your body would make polio antibodies, and that's how you've become immune to polio. That was the original Salk vaccine. That's what Jonas Salk did. Jonas Salk took live polio virus, attenuated it, broke it down, denatured it, injected it, and voila, you had immune competence against polio, so nobody got polio anymore. That vaccine, that methodology is also used for tuberculosis. The BCG vaccine that has been around for 80 years, or 70 years, I should say. They would took the bacillus, they would take the tuberculous bacillus bacterium, they would boil it down, denature it, inject that into your body, and then voila, you were immune against TB because your body would see that product, make antibodies, little proteins against it, so that if you did get coughed on by somebody with TB, you wouldn't get TB because you've already seen components of that virus before. So that's called the inactivated pathogen strategy. And the issue with that is it's tried and true. We can take the coronavirus and boil it down and denature it and throw it in some alcohol. We can do all of that, or we can grow variations of the coronavirus in chicken eggs and pull out those attenuated vaccines, and then we can inject that, and then you can be immune to coronavirus. Problem is, typically these types of strategies rely on chicken eggs. So if you have a chicken allergy, forget about that. And secondarily, this is an extremely slow process. So remember, we're looking at a scale that is going to require billions of doses. So to grow billions of doses of inactivated coronavirus pathogen in chicken eggs is not going to be a very feasible scenario. But again, people are working on it. The Chinese, number one in that regard, and the Australians are number two in that regard. And I want to finish up by saying there's a lot of politics involved in all of this, because remember, we've talked about the Chinese, we've talked about the Australians, we've certainly talked about the Americans, we've talked about people in my home state of Maryland, we've talked about the Brits, we've talked about Sanofi, the French. So you say to yourself, wow, this type of vaccine development seems to be occurring all over the world. Why is that? Well, it's because many countries, the United States included, have things called Defense Production Acts, which means that if we were to develop a vaccine here in the U.S., we might want to swoop down and keep all of that product for American citizens and not export it. Well, that type of thinking is also occurring in the EU, it's occurring in Australia, and it's definitely occurring in China. In fact, I told you that the viral vector component is a division, that vaccine development is from CanSino, a division of the People's Liberation Army. So make no mistake, there is an extraordinarily nationalistic component to vaccine development, because we are talking about what is the equivalent of the moonshot. Whoever finds the vaccine that works, it's like landing the first man on the moon. So every country that we've talked about today, Canada as well, every developed first world country is in the business right now of utilizing one of these 5 strategies to come up with a vaccine that they may first deliver to their own citizens and then sell on the open market. Well, obviously, that creates another political discussion as, well, if you only inoculate your own population and somebody lands in Kennedy Airport from Milan, Italy, and they haven't had access to the vaccine, well, what's that now mean for the population in New York? So we have to approach this in many respects on a global basis, but you've got a lot of these companies that are based in other countries, or they have multiple locations, meaning they have a plant here in the US, they have a plant in Europe, and they also have a collaboration going on in China so that they can cover all of their bases, because make no mistake, whatever country finds the vaccine first is basically the first country that has landed on the moon. So there's an extraordinary nationalistic push to get these vaccines developed. So let's recap where we were. History of vaccination, going back to Edward Jenner, taking cowpox and putting it into people to protect against smallpox. That's how we get the term vaccination. We've got different FDA phases that have to be achieved that will at least take 1 year to bring a vaccine to commercial market before you or I will see that vaccine. And then we've got about 5 strategies for vaccine development. We're looking at viral vectors, where we are using other viruses to get components of the coronavirus into our body so that we can mount an immune response. We've got the RNA strategy, where we're taking ribonucleic acid, messenger RNA from the coronavirus, injecting that into the human, having us make lots of spike proteins that will then initiate an antibody response so that we're now immune to a coronavirus when Seymour at the table over there doesn't wear a mask and starts to hack on my corned beef sandwich. And then we've got strategy number 3, which is the Inovio strategy, which is DNA, meaning they're taking the DNA interior of the coronavirus, injecting that, and generating a neutralizing antibody response. So that's strategy 3. Strategy number 4 is taking viral proteins, taking bits of the crown, bits of the sphere, injecting that, getting an immune response so that when I go bowling next to this guy who's not wearing a mask and he coughs on my bowling ball and I wipe my eyes, that's now going to protect me because I've now got the viral protein vaccination. And again, we've seen this historically. Human papillomavirus, flu shots, this is where that technology comes from. And then, of course, we've got strategy number 5, which is just inactivated pathogen strategy, taking weak forms of coronavirus, injecting them into chicken embryo eggs, letting those eggs make those viruses, extracting all of that out, and then injecting. That works in flu, it works in BCG, it works in polio, it meets a lot of resistance from people who have egg allergies, and it's a really, really slow process. So I hope you learned a lot with today's discussion. I've given you an overview of how the 5 different vaccine strategies are in place, going back to Edward Jenner, which of course was kind of like inactivated or similar pathogens. So we've used these strategies since the 1780s, 1790s, and it has served mankind very well. We're looking at a moonshot here, meaning this is the race for the moon. It is achievable. I do believe that we are going to reach the moon. We are going to get a vaccine. We may have multiple vaccines using one of these or all of these 5 strategies. And remember, this is a real nationalistic endeavor. So the first one to come up with the product is going to reach a significant holy grail. But you're looking at companies that have a foot in China, a foot in the US, a foot at Oxford, a foot at Imperial College, a foot in Canada. So it's a very multinational endeavor. And then, of course, the big question is going to be, what are you going to charge for it? Some folks are saying maybe $30 a shot, but that's pretty good money when you multiply that times 6 billion humans. And other folks are saying, well, maybe these companies will be able to market on a multinational basis. So you're going to have to do your homework when it comes to seeing which companies. But I can tell you right now, it's probably not worth investing in any of them, at least until we get more phase 3 clinical trial data. And I want to remind you, SARS has been around. Which is the parent virus of coronavirus. SARS has been around since 2000. MERS has been around since 2013. We ain't got vaccinations for them. Maybe there won't be a vaccination for coronavirus, COVID-19. Maybe there won't. There's a lot of money invested in this, billions of dollars. A lot of brilliant minds are looking at this very issue right now. So in my heart of hearts, I do believe that we will have a vaccine. And then, of course, we're going to have a conversation with the anti-vaxxers who are going to say, nope, I don't want nothing foreign in my body, except for the fact that every day of your life, you have something foreign in your body. So on that note, I want to thank you all. I hope you learned something today. Take your envelope, put it in your pocket. And if you go out to dinner with somebody wearing your mask and wearing your gloves, maybe you'll have an interesting conversation and you can use the knowledge that you learned today to kind of deflect all the misinformation that's out there on social media. So thank you. Stay healthy. I'm sending you a big virtual hug and an air kiss. Have a great day.